Good afternoon, and thank you all for joining Scripps Research and the Mind, Music, and Movement Foundation for Neurological Disorders. I'm Beth Elgort, the founder of Mind, Music, and Movement, and I am so thrilled to be here today with Dr. Srinivasa Subramaniam and Scripps Research. How lucky you are to be in the comfort of your own home, thanks to COVID and Zoom. Positive spin. Sit back with your beverage and snack of choice and enjoy our time together. In 2018, I was blessed to have shared a vision with two wonderful ladies, Andrea Finkel and Arlene Cohen, representing the Frenchman's Creek Women's for Scripps Research. Tonight, we are dedicating this event to Arlene Cohen, who has since passed. It was her passion to have Scripps Neuroscience gain funding for research in Alzheimer's, along with our vision for all neurodegenerative diseases. We are doing just that. Arlene, I know you are smiling from above. 4 years ago, I started my mission to bring resource awareness for Parkinson's and neurological disorders to Palm Beach County. Thanks to Scripps Research and my visit with Arlene, Andrea, Dr. Ron Davis, Sue Rohde, and Tara Holcomb, we hosted our first symposium. It was Scripps Research and their more than amazing staff, Jen Beatty, and everybody there that helped us make this happen. We had standing room only. Over 250 people attended. Dr. Corinne Lasmanis educated us all with her wonderful information about Parkinson's, along with presenters and vendors. I guess in research terms, Scripps made it possible for us to test our hypothesis that there was a need for us to serve our community. April 11th, 2018, we gave birth to our foundation's vision for the future. Our foundation is about keeping the mind, body, and soul fluid. We support our community with amazing resources to help families living with neurodegenerative diseases with our programs, classes, events, and resources. Today, we are for now virtual through our website. Our hope is to help people delay progression of the disease, and when we have the needed funding, build out our center and donate a percentage of our funds to Scripps Research, taking steps forward towards a cure. You can visit us at m3f.org. I realized immediately after meeting the scientist I'm about to introduce, he is one extra special person. All I keep thinking about is the television show Big Bang Theory. He has the warmth, heart, and brains, along with a great sense of humor that just make you smile and feel tremendous respect. I will tell you why with a brief story. He grew up in a rural town in southwest India. His passion for science was clear. He earned his way to pre-university, helping his father sell vegetables and betel leaves to the local markets on Sundays, saving every rupee he earned. He continued from there to the top. He earned his doctorate graduating summa cum laude from the University of Heidelberg, Germany, and did his postdoctoral work with Johns Hopkins before arriving here. His education, training, awards, and honors speak major volumes. He started a 501c3 with 8 other friends while in college to help underprivileged children in a rural southwest India have educational opportunities and leave poverty behind. He understood this only too well as a child. Every year, this trust, which is a 501c3 nonprofit organization, supports hundreds of children, providing them with financial support, arranging science seminars and science experiments in competitions in rural schools. In his neuroscience lab, he directs a team of researchers delving into the mysteries of the brain. They are investigating how diseases such as Alzheimer's destroy the brain's neurons that transmit thought, language, movement, and memory. then it might be possible to develop drugs to halt the process. Tonight's conversation is tunnel-like communication in the brain. You will be muted and can raise your hand and write questions through chat. You will be unmuted to ask questions for Q&A at the end. So without further ado, I give you this very special man, Dr. Srinivasa Subramaniam.
Thank you. Thank you. Appreciate that kind introduction. I'm really thankful for this opportunity, and I'm very glad to be here and share some of the work we are doing at Scripps. And it's wonderful to interact with communities and share our research. I really appreciate Beth, Georgia, and Tara for arranging this. I hope I would meet all of you one day after the COVID in person. Yes, so today I would like to discuss with you some of the interesting work that we're doing. Particularly, I would like to discuss with you a very interesting communication that we found between the neuronal cells in the brain. So communication is, we all know, is very important for human. That's why human is slightly different from other species, because we can communicate through many different ways. And this communication is very, very old for us, and which made a very important steps for our evolution, our progression as a species. So not all communications are conducive. As you know, some communications are very friendly, but some communications could be very not so good. So it's not very friendly communication. So, and we know that communications is very important, even in the modern time, that through communication we interact and express our feelings and opinions. So evolution of communication, if you look at, we actually communicated using symbols before, thousands of years ago. But now we're still doing that. Currently, we're doing the same thing. We're communicating through symbols, which is kind of, I found very interesting, that our communication has been, even though primordial, but it acquired a kind of digital shape for it. Why does we communicate? What is the need for it? Communication is big. We communicate because of our brain. Our brain is evolved, and it has many different regions, and which controls many different functions. And through this function of this very basic structure in our bodies, brain is the cause for all our communication. So if you want to appreciate the importance of communication, you can look at the developing brain. Here is the cartoon structure of my daughter when she was small, like 2.5 years old. This is how she imagined the human face. When she's growing, like 3 year and 4 year, she gradually acquired these different shapes, how a human, the feature of human. So this happens because the developing brain acquires this information from the outside and start computing and then expressing. So the communication becomes more and more matured when the children becomes when the children are developing. So when this particular that's this basic function of the brain, when it's affected, as we know, the brain is affected by many different diseases. So when this fundamental feature of the brain is affected, then we end up having many different kind of problems. One is neurodegenerative diseases, and the psychiatric and mental disorders, as well as developmental disorders. So if you look at the brain is not composed of one kind of cells. There are many different kinds of cells. They are present in different locations and many different shapes. As you can see here, there are there are many different types of cells in the brain, and they communicate in a way that even a simple task that we assume is granted, like drinking a coffee or just taking a walk or watching a television, all these are regulated by this fundamental set of cells which are working properly in our brain. So if you look at, like, for example, one neuron from the brain, here is a neuron from the hippocampus, which regulates memory. If you look at this beautiful structure, which has many different kind of protrusions on the left and the right side. And through the end of these protrusions, these neurons interact. If you zoom inside, you can see this is the ultrastructured details. If you zoom in further, you can see these small spots of this interaction. These are called spines. So neurons have these enormous complicated structures, and they are made to interact with another neuron through this end called spines, which is present in each neuron may have a multiple spines, thousands of spines. That's how they interact with the other kind of cells. What we know about how neurons communicate has been a study from like 100 years. We know from 100 years that neurons communicate, and the two different major communications signals are chemical signals and electrical signals. This is we know very well from a long time. What we discovered in the laboratory is that a unique form of communication, which we call tunneling communication. There's a form of tunnel formed between the cells, and through those tunnels, the neuronal cells communicate each other. Here is a neuronal cells. I'm going to run this video. You can see how these neurons are talking to each other through these long protrusions. This I went a little bit further zoom. You can see that neuron has this long protrusion, and these are the green spots on the other cell. And after some time, you can see that this is we call cargo coming from the green cell, entering into another cell. So this way of communication, we call them as tunneling nanotubes. And you can see those communication that this kind of tension on this communication, and they can start delivering the vesicles, vesicular punctures or cargoes into the other cell. So if you just get an analogy, this is like a tunnel between two cells and transporting cargoes. I wanted to show you another video where these two cells are connected with this tunnel-like structure, and they're transporting cargoes. Here, there's a cargo called endosome has been transported. So this is very interesting because this is the first time that we have shown that there is this kind of communication in the brain, which generates a lot of questions about like what are the functions of this kind of structures. So we did also high resolution imaging. Here, what you can see is there are these two cells here, one cell 1 and cell 2. And they can see this is the tunnel protrusions. They're coming out of this cell. And they seem to have some kind of bifurcations from these protrusions. And they start interacting with the neighboring cell. So coming back to our major question, okay, we discovered this new form of communications in the brain. Okay, what does it do? What is the use of these tunnels in the brain? So this is a question that we would like to answer. So as I mentioned, there are three major diseases, neurodegenerative diseases, which has no cure. A normal brain can turn into an Alzheimer's brain, or it can turn into a Parkinson's brain or Huntington disease. We do not know why different people get different kind of diseases. Although there is some genetic component, we still don't understand why these diseases affect different parts of the brain. Like in Alzheimer's disease, the hippocampal and internal cortex is affected. In Parkinson's disease, substantia nigra is affected. In Huntington's, the striatum affected. So each disease has a different region, anatomical specificity, and they have a different symptoms. So the very fundamental feature of the brain, like remembering and walking and all these are affected when we get these diseases. So what we have found over 10 years now is that we are working with there's one important feature of all these different neurodegenerative diseases is that the proteins involved in these diseases, they all form aggregates. They change their conformation in the brain, in the cells, and they start aggregating and affecting the normal functions of the cell. And it has been known for many, many years, like from past 10, 15 years, that these disease-relevant proteins, they move from one cell to another in the brain. They move from, they make connections, they go in the form of vesicles from one cell to another. But we still don't know the details of how this kind of cell-to-cell movement of this disease-relevant protein occurs in the brain. So what we have been working on is that protein called ResRAS homolog enriched in the striatum. What we have found is that this protein is highly enriched in the brain region called caudate putamen, the region which is involved in Parkinson's disease, and hippocampus, the region which is also involved in Alzheimer's disease, and also to some extent in the cortical areas of the brain. So, and this protein is this is the mouse brain. You can see in the human brain also this protein is highly expressed in the brain. So, from past few years, there's studies showing that this protein RES is required for tau pathology in Alzheimer's disease. And we showed that this RES protein is required for motor abnormalities in Parkinson's disease. And also we showed that RES is important for Huntington's disease pathology. So these studies have been published. Due to the time, I'm not going to details of this study, but the bigger picture is that the RES protein can form these tunnel-like protrusions in the brain. As you can see here, this is the three-dimensional structure where RES is expressing in one set of neurons. And if you can see, these are the protrusions in the brain that connecting to another cell, which is red here. So when the red and green becomes together, you can see yellow color. So this data shows that the RES can form these protrusions in the brain and probably transporting some of the disease-relevant proteins. Here is an example. What you're seeing here is RES making these long protrusions between neuronal cell and transporting neurodegenerative disease protein. You can see here, this is the vesicular structure carrying the disease protein to another cell, from one cell to another. And you see, this is the fragile tunnel. This is very, very fragile tunnel, but it's kind of holding onto these two cells and transporting the disease-relevant protein. Here is another example I want to show you here. You can see that this tunnels is more like they are different labeled because they consist of RES itself and also disease protein. Disease protein is in red. If you can see, this tunnel kind of punches, makes a hole in the other cell and delivers this cargo. You can see this is the green or the yellow or the red. They're the cargoes that have been delivered from other cell. If you express this protein in one part of the brain, for example, in the region called striatum, and you can start seeing them in other part of the brain after some weeks. You can see here. We injected this protein here, but then we start seeing them on the other part of the brain. So, so there is a movement of this protein from one region to another region, indicating that this is something that has been not well studied before. We think that this kind of transport can occur because of this tunnel-like transportation. Here's another example. I want to show you that these two cells are connected and they're transporting the neurodegenerative disease associated Huntington protein from one cell to another. So I showed you some data about the relevance of these tunnel-like structures in disease context, like neurodegenerative disease context. Here is another video I'm showing you that this is the protrusions and they're interacting with another cell and they're delivering the cargoes. And this kind of interaction that occurs right now when we're talking, this kind of interaction may be occurring in our brain. And we think when these kind of interactions are not properly regulated, then we may end up having brain diseases. So we think RES tunnels also might be involved in psychiatric diseases, because if you have a mice, if you have a RES knockout mice, they are hyperactive. So here I'm going to show you the wild type mice. That wild type mice is on the left side, RES knockout mice on the right side. What you can observe is that the wild type mice moves in this chamber to some extent, but you can see the RES knockout mice are moving at a higher rate. So they're kind of constantly moving. They're hyperactive. So when you have this protein which makes this kind of tunnel-like structures in the brain, if we do not have those structures, we presume that the RES knockout, those structures may not be there. So that results in, for example, hyperactivity. So our thinking is that if you can somehow modulate these tunnels in the brain, first we understand them mechanistically, okay, what they are doing in the brain and what they're transporting. We showed that they transport Huntington disease protein, and also we think that they also transport tau proteins. If you can modulate those transport, if you can prevent, for example, those kind of transport, we can presumably prevent some of the disease aspects. For example, RES is very critical for L-DOPA-induced dyskinesia in Parkinson's disease. So if you can target RES, we think that we can reduce L-DOPA-induced dyskinesia. So we are currently trying to understand how this kind of tunnel might be involved in Parkinson's disease, especially in the movement, because the RES knockouts are hyperactive, and we think that when there is the lack of communication, proper communication between the cells mediated by RES, that might lead to some of the symptoms that we see in neurodegenerative and neuropsychiatric diseases. So our goal is to understand these structures more and develop drugs to go forward. So here is my lab. And I would like to thank the research funding agencies who support our research. And here is my team. And I would be happy to take any questions or any comments. Thank you.
So Doctor, thank you very much for sharing your presentation with us today. I am Tara Holcomb. I am the Senior Director of Philanthropy at Scripps Research, and we truly appreciate Beth Elgert for hosting us today, as well as your presentation. There are a few questions that will be coming in. So the first one that I'm seeing on our screen here is, what is causing these things to happen in some brain regions and not in the others?
Yeah, that is the question we don't know. Maybe, as we all know, that different regions control different parts of the body, and some regions may require this kind of communication. So we still don't know which, if you're talking about the formation of tunnels, we think that this kind of formation can occur in any part of the brain.
Okay.
But if the disease onset, why the disease occurs in one part of the brain, we still don't. We're still trying to understand those complexity, and we think that there's the mechanisms in those regions, affected regions, which are controlling the disease onset and progression. And our lab is particularly interested in understanding those mechanisms.
Okay. The next question is from Dr. Sherman, and he is asking about the tunnels specific for each substance traveling through the tunnel. And are there different tunnels for different proteins or neurotransmitters?
It's a very good question. It's a great question. These tunnels, we think, right now we are looking at the resolution of 100 nanometer, say 100 nanometer resolution. To answer the doctor's question, we need to get into a resolution of like 20 nanometer. So, and we are in that difficult situation to really get into that level. And moreover, these structures are very fragile. Like, you have to treat them very gently to know those. I think to answer this in the simplest way, we don't know exactly. We think that there could be different tunnels depending on the cargoes that have been transported. Or it's a general tunnel, like a tunnel between two mountains. Anything can grow, but you see most of the tunnels have limitations. You cannot take a big truck or a big truck into the tunnel, for example. So we don't know. I think those are very important questions. We are very excited to address those questions.
Yeah. Do we know what these tunnels are made of?
They're membrane. They're made up of membranes, plus plasma membrane and proteins component. So the whole structure is made up of membrane. We know that it's membrane because we have multiple evidences. So, and proteins. The building blocks of the tunnels are all membrane and the proteins, which kind of hold on to those, make those tunnels along this.
And once they make the tunnels, are they permanent, or do they just appear when needed to transmit, or are we stuck with them for the duration?
This is a very good question. So the thing is that based on looking at so many videos in this the transportation, we think that the tunnels have purpose. They transport cargoes. Once the delivery is done, they retract. They retract back. So I think these tunnels are formed, and then once the delivery is over, there is something says, okay, done. Delivery is done, then go get back. So we think it's like that. Like, it's like a postman coming and putting the post and then going back. There is no constant, there is no permanent connection there.
Okay. Now, what does your team use to visualize? You had some great illustrations in the PowerPoint that you shared with us, but how difficult is it to see these tunnels and them transporting like the cargo across? Is it through microscopes, and what type of microscopes do you use?
Yes, so human eyes, we cannot see at 100 nanometer resolution. We can't see bacteria, for example, right? We can't see them with our naked eye. We need microscope. So these tunnels are, as I mentioned, are 100 nanometer structures. You need a microscope, high resolution microscope, to visualize them. So we always use high resolution microscope, and we do most of the time live cell imaging. That means we just see the live cells that are alive, and so we capture those images using microscope.
Okay. We also have an attendee who was recently prescribed with a booster to the carb level because their walking got worse. They've been diagnosed with Parkinson's and they're not feeling any better, feeling dizzy, not well, and they're looking to get off of this. Is there any type of therapies out there to help with symptoms, or therapies that are in the pipeline to help people with these neurodegenerative diseases?
So here is the, Tara, I missed you the first part. You said the patient was on what drug?
They just say that they're on a booster type drug to help with their symptoms. So we know that there's medications for symptoms out there, but do we have any others that are in the pipeline that either your team are working on or Scripps is working on, or they're that we're not aware of?
Yeah, so what I know is that if they're considering taking a levodopa, which is a gold standard drug, that most of the Parkinson patients receive a levodopa. It's also called L-dopa. So currently, depending on the severity of the symptom and the dose, how severe is the echinacea, the neurologist would decide what to give and what not to give. So for most of the Parkinson disease patient, the levodopa is the gold standard drug. But unfortunately, not every patient reacts favorably. They may react for some time favorably to the drug, but then the drug kicks in a side effect, which is levodopa-induced dyskinesia, which is a major problem. So I think right now in Scripps, we are trying to develop alternative drugs, for example, blocking RES, or we are developing and we are understanding another molecule called RasGRP1, which is potentially a great drug, we think, for Parkinson therapy. So yes, we are under different level to really get into this drug discovery research on Parkinson disease therapy. But as far as alternative therapies, existing alternative therapy, I'm not aware of those any alternative therapy apart from levodopa.
Okay.
You can begin giving. Yeah, I'm not aware of.
Okay. And you were speaking about punctures of the cells, so to transmit the proteins across. In the COVID era, we're seeing a lot of conversation about the spike-like proteins on that cell. Do you see something similar when you're talking about puncturing? How is that puncturing happening? Is there anything that you guys are able to visualize?
Great question. Why I'm telling this is another important and great question is because we have to know like how this protrusions, it has to hang on from the cell for some time, then looking for a neighboring cell. Once it finds a neighboring cell, it has to interact with that cell. I said it's kind of poking, making a hole, because then only it can deliver this vesicular cargo. There is no way it can deliver with when the gate is closed. Vesicular cargos are sometimes like 20, up to 20 nanometer thickness. So it has to open up the acceptor cell to deliver this cargo, and how that it happens and what is that it poking through what proteins, we don't know.
Okay.
So those are the next questions that we want to address. We are so excited at this point, like why it is formed, how it is sustained, and how it is delivering, how it pokes the cell. There must be, we think there must be some kind of coordination from the acceptor cell and the donor cell for these protrusions to open. So we don't know those details yet.
Okay.
Great.
Well, hopefully maybe we can get together next year and you'll have that answer for us.
Yes, I look
That would
forward.
be great, wouldn't it? So what about, we have a question about early detection for diseases like Alzheimer's, Parkinson's. Is there any guidance to the attendees today when they're feeling they may be symptomatic? Is there options for them to seek early treatment or to get answers that they have that type of neurodegenerative disease?
Yeah, so again, I'm not a neurologist, I should say. These things are evolving. There is a new therapies, and there is a big companies, and there's a new drug that has been delivered to the market. And the best option is, the best way you can know about those things is your physician, the whom you're interacting with. And unfortunately, as you know, that the Biogen drug, which a couple of days ago, which is made to prevent amyloid beta toxicity, has been kind of not been approved because the amyloid therapy was not successful so far. So there is a lot of effort, as you know, there's a lot of effort on this field. I think the best person is the physician that whom they are interacting with, to know about the details and the latest drugs and all this. But currently, I should say, I'm afraid to say, we do not have a strong predicament like how to diagnose this disease and how to cure them. It's all, every patient is different.
Correct.
There is no homogeneity among Alzheimer's disease patients. So I think the best person to interact with is the physician that they're talking.
Okay, yeah, great advice. Always seek your medical professionals for that stuff. Yeah. So we do have Sue Carroll who would like to answer you ask you a question directly. So we're gonna give you permissions, Sue Carroll, just in a second here to ask your question.
Sure.
I just said thank you because I am the patient that has the booster they gave me to the carbidopa levodopa, and it does not seem to be working. As a matter of fact, I seem to be worse with my walking, which is my main terrible symptom. And I realize that other people have a lot of worse symptoms than I do. But my question is getting off of this new drug that she put me on. I am reticent to get off of it, but what is your opinion? I know that you're.
Yes, thank you, Sue, for contacting me. I think what you mentioned is that levodopa is not working for you.
No, the booster is not working for me. I've been on levodopa for about a year, and she just put me on, my neurologist just put me on a different type of drug to boost the levodopa, and it doesn't.
What they do is that, as you probably know, they're trying to keep the level of levodopa in the brain by inhibiting another component, so that they want to modulate the level of levodopa in your brain so that it acts in a way that it will help you in your movement.
I'm interested in the blockers that you talked about, that you're working on some blockers that actually will more or less stop the bad cargo from going in.
Yes, so we think we have a protein which makes which is involved in this kind of movement dysfunction, but we are currently do not have a small molecule like a drug, like levodopa, like something, that kind of drug to block that protein. So we know we have a candidate, which is a great candidate. We can block it and prevent some of the motor deficits. But right now we are in a stage that we need to develop, screen for drugs to block this protein.
Mhm. Is there any way to get into a clinical trial for any of this?
No, we are in the very at the first stage of discovery, which we would like to make. Like we wanted to we first need to figure out what drug is actually functioning. And then we go to a next level. We have a great preclinical models. We have a macaque model, we have a monkey model. We're collaborating with a French group for macaque model. So we have a great team to test this compound. So we are currently looking for grant support from NIH and other private foundations to really screen for the small molecule drug for this protein. So then we go to the next step, preclinical model, then we go to the clinical side. So we are in the beginning, unfortunately, on that. So we need more support to go there at the clinical level.
Thank you.
Thank you.
So Carol, I also bring up a great source, if your physician has not indicated to you, is clinicaltrials.gov. So anybody that is looking to participate in clinical trials should always work with their physicians, so their doctors that are treating them, and see if they're candidates for any clinical trials out there. So just like anybody at Scripps, like our scientist today who's speaking, or others, once they get to the clinical trial phase, they'll be posted there. So it's a great option for you to look into. So I do not see any other questions, but I will give it just a couple of seconds in case there's some other items that are lingering out there that anybody is getting to type in right this second. Is there anything else, Doctor, that you want to share with the attendees today about your science, advancements, next steps?
No, appreciate any support that they can give us for the research support. Be great.
What type of resources do you use in your lab to advance your science, or where do you get your funding from?
Like right now, we get most of funding from NIH and also from private foundations, like in this case, the CHDI Foundation. Also, we got some money from community foundations who are supporting $25,000 for us. So we get many different sources to get this research going. Yeah.
Great. Well, thank you so much. I do not see any other questions coming up in the Q&A session. So at this time, unless Beth would like to come back on and wrap us up, that would be great.
Thank you all so much for participating tonight, and a huge thank you to our presenter, Dr. Srinivasa Subramaniam, and to two very special ladies, Tara Holcomb and Georgia Ringler. Our relationship with Scripps is treasured. We hope you all will continue to support all the fine work Scripps Research does now and in the future. If you would like to learn more about us, you can visit our programs, and we're offering these wonderful virtual classes and also webinars. And we have an upcoming symposium January 23rd. So we are at m3f.org. And Tara and Georgia, you're awesome. Srini, mwah. Thank you all so very much.
Thank you, Beth. Appreciate it.
Thank you, Beth, for helping us.
Thank you.
See you all.
Hope everyone has a great evening.
You too.
Great evening. Thank you for everybody. Thank you.
Bye-bye.